This slide looks at the difference between two types of diabetes. Diabetes mellitus is a disease when you cannot control the blood glucose, and there are two principal types of diabetes. Type 1 diabetes, which is also known as insulin dependent, and it's sometimes known as early onset for reasons I'll describe in a second, and as type 2 diabetes, which is known as non-insulin dependent and is sometimes known as late onset. Okay, so diabetes mellitus is an inability to control blood sugar levels. Either you can have blood sugar levels which are too high, which is known as hyperglycemia, or you can have blood sugar levels which are too low, which is known as hypoglycemia. The symptoms of those are summarized here. So hyperglycemia, high blood sugar levels, results in an extreme thirst, dry mouth, sometimes blurred vision, drowsiness, and a frequent need to pass urine. Hypoglycemia, which is low blood sugar levels, feeling shaky and irritable, sweating, tingling lips, feeling of weakness, hunger, and nausea. Okay, so those are all the basic symptoms. In terms of the differences, if I go back to the top here. Okay, so the type 1 or insulin dependent or juvenile onset, because it tends to happen in young children or develops in childhood, and it's a result of the patient not being able to produce insulin. Okay, and it's often caused by what's called an autoimmune disease, which is when one's immune system attacks its own cells. Okay, so in this case, the immune system is actually attacking the beta cells in the eyes of Langerhans, and therefore they're unable to produce insulin. Sometimes it can also be caused by a viral infection. It's a thing that can happen post-viral infection when the viruses have obviously attacked the beta cells too. The treatment is injecting the patient with insulin at mealtime, so just before the blood glucose levels is about to rise due to feeding, you inject yourself with insulin, which controls the potential rise in blood glucose. Okay, and whereas type 2 or non-insulin dependent happens later in life, more prevalent in people over 40, hence it's known as late onset, and this results that insulin is produced, okay, but the target cells don't respond to the insulin. Okay, so the receptors and the transduction cascade inside the cell doesn't work, and therefore the insulin doesn't have the effect on liver cells and muscle cells and fat cells, so the after a meal the blood glucose doesn't fall despite the production of insulin. Okay, there are certain risk factors for type 2 diabetes. Some of them are lifestyle, some of them are genetic, so obesity or a high sugar diet or excessive alcohol consumption would be lifestyle factors which add to the chances of you developing type 2 diabetes later in life. But there is also genetic predisposition. Okay, if it's common in your family, you'll have a higher chance of also getting type 2 diabetes, and it's more common in Asian and Afro-Caribbean communities than normal, well, not normal, but European communities that we encounter in UK. Okay, now the treatment of this, obviously injecting with insulin isn't going to work, because the insulin is the amount of insulin is not the issue. It's the fact that insulin doesn't work. So the only way they can really control this is by controlling diet and exercise, and balancing diet and exercise to control blood sugar levels. There's some diagrams and animations to illustrate all of this. So this diagram here, very simple diagram, shows the difference between type 1 diabetes. Okay, so we have high glucose levels in the blood, but no insulin to bind to insulin receptors to be able to allow these glucose molecules to move into liver cells or muscle cells or fat cells. With type 2 diabetes, we have the glucose here, but we also have the insulin here. But the insulin, whilst it may bind to the cell surface receptors, there's no response inside the cell. Okay, so the those glucose transporters aren't inserted into the cell surface membrane, and therefore the glucose can't move from the bloodstream into the liver cells. Again, another diagram which might be more helpful. So this one here deals with a healthy individual. Okay, so they can produce insulin, and that insulin will bind to target cells such as the liver, which then place glucose transporters, GLUTs, into the cell surface membrane, which allows glucose into cells. As a healthy person, type 1 diabetic, their problem is that they don't respond. They don't produce insulin. Okay, so no insulin, and therefore, if there's no insulin, that can't bind to the receptor. If that doesn't bind, if nothing to bind to the receptor, the glucose transport molecules remain inside the cell, and glucose can't get into the liver cell. Whereas type 2 diabetes, they produce the insulin, but the insulin either can't bind to the receptor, or the signal transduction cascade doesn't work inside the cell, and therefore in either of those situations the glucose transport molecule doesn't fuse with the cell surface membrane, and glucose can't get into the cell. Moving down to look at some animations of this.
Diabetes affects the pancreas, an organ of the digestive system. An important function of the pancreas is to make hormones that regulate blood glucose levels. Insulin is produced by beta cells, which cluster in groups called islets. Insulin is a hormone that lowers blood glucose levels. After a meal, glucose reaches the pancreas via the bloodstream and is released from capillaries into the pancreatic tissue. This triggers the release of insulin, which is stored inside the beta cells. Insulin travels to other parts of the body via the bloodstream with three main destinations: muscle, fat, and liver. Insulin helps these tissues to store glucose as an energy source. Shown here are muscle cells. Insulin arrives through the fine capillary network that traverses the muscle fibers. The surfaces of muscle, fat, and liver cells are covered with insulin receptors. When these receptors bind insulin, they signal to the cells to release special glucose transport channels onto the cell surface. These channels enable glucose to gain entry into the cells, allowing the cells to store glucose as energy for future use. In this way, insulin acts to regulate blood glucose levels. In type 1 diabetes, a patient's own immune cells mistakenly attack the insulin-producing beta cells. Type 1 diabetes is referred to as an autoimmune disease because the body's immune system attacks its own cells in error. Over the course of the disease, a patient's immune cells will systematically but mistakenly kill nearly all the beta cells, shrinking the islet but leaving all other cells intact. The few beta cells that remain in the pancreas have a severely reduced capacity to produce insulin. The death of the pancreatic beta cells and the consequent inability to produce insulin is at the core of type 1 diabetes. Without insulin, the body cannot regulate blood glucose levels, and the high glucose levels damage many tissues throughout the body.
And moving on to look at type 2 diabetes, I've got another animation here.
Understanding type 2 diabetes. Type 2 diabetes is a metabolic disorder that causes sugar, in the form of glucose, to accumulate in the blood rather than being used as fuel by the cells in our body. When we eat, food is broken down by our digestive system into nutrient molecules that are then absorbed through our digestive tract for use by the body. Foods containing carbohydrates or various sugars are broken down into glucose. Glucose is an important source of fuel for many organs in our body. However, to be able to use the glucose for fuel, the glucose molecule must first enter into the cell. The pancreas produces a hormone called insulin, a chemical messenger essential for the entry of glucose into cells. As the blood glucose levels rise after a meal, insulin is released into the bloodstream and sets processes in motion to trigger the removal of glucose from the blood to enter into the cells. In type 2 diabetes, the cells become resistant to insulin and ignore its message to absorb glucose. This is known as insulin resistance. In addition, in type 2 diabetes, the pancreas is unable to produce the greater amounts of insulin needed to trigger these resistant cells to take in glucose from the bloodstream. The most notable symptom of diabetes is frequent urination and excessive thirst. Other symptoms include weakness, drowsiness, and blurred vision. These are caused by chemical imbalances in the blood related to high levels of blood glucose. About 1 in 4 people with type 2 diabetes are unaware that they have the disease. It is important to catch diabetes early. Over time, high blood glucose damages the blood vessels, which can damage the organs that these vessels supply, leading to a variety of health complications. Damage to the small or micro blood vessels can cause vision problems, including loss of sight, nerve damage, and kidney disease. Damage to larger or macro blood vessels can lead to cardiovascular complications such as heart disease, stroke, and poor blood circulation. Overweight and inactivity are major causes of diabetes. A family history of diabetes significantly increases your risk of developing the disease. Certain ethnic populations are also at increased risk of developing type 2 diabetes. Finally, some medications may increase your diabetes risk, specifically corticosteroids, thiazide diuretics, drugs used to treat certain mental illnesses, and some antiretrovirals used to treat HIV infection. In summary, type 2 diabetes is a metabolic disorder. It causes sugar, in the form of glucose, to accumulate in the blood rather than being used as fuel for the cells in our body. If not diagnosed and treated in a timely manner, type 2 diabetes can lead to many health complications.
So just very quickly, sorry, this slide is all about the differences between type 1 and type 2 diabetes, and so hopefully that illustrates that, and hopefully these summary notes are useful. Moving on to the last slide in this screencast. This discuss the use of insulin produced by genetically modified bacteria and the potential use of stem cells to treat diabetes mellitus. Okay, so type 1 diabetes can be produced by, or can be treated by injecting insulin into patients, and traditionally, and that insulin was isolated from either horse or pig blood, and was isolated, packaged and sold to diabetics. Nowadays, what we're able to do is to take the gene for producing human insulin out of a human being, insert it into bacteria, and produce insulin in that way by fermentation. And we'll look at the process of how that is done in a lot of detail when we move on to F214 next term. But also there are a few advantages of using human insulin over animal products, and I'll very quickly go through some of these. So human insulin is far more effective than porcine or equine insulin, porcine being from pigs and equine from insulin. It's an exactly the same molecule than the insulin your beta cells would produce, and therefore it fits the cell surface receptors more effectively, and it seems to work more quickly as well than either porcine or equine insulin. Because if you inject proteins from other organisms into, there's a risk of immune response. You might recognise the porcine insulin or the equine insulin as being non-self and have an immune response to it, which would be dangerous. We have issues of infection between animals. I've got prions here. Prions being the proteins which cause diseases like BSE in cattle, or Creutzfeldt-Jakob's disease in human beings. So there's a danger of using animal products, blood products, that you could get those. And also there's a huge demand. There's an awful lot of type 1 diabetics around, and producing insulin by fermentation can produce lots of insulin very easily, as opposed to having to bleed horses and pigs and isolate the very small amounts of insulin that would be present in their blood. Is a very time-consuming business, and wouldn't necessarily produce very large amounts of insulin. And also there is various religious things about using, or vegetarians wouldn't want to inject themselves with porcine or equine products, and then obviously with porcine products we have various religious issues with people wouldn't be willing to inject themselves with any form of pig products too. Okay, now whilst this is a type of treatment to alleviate the symptoms of diabetes, it's in no way means a cure. However, there is the potential for a cure by the use of stem cells. If we could find stem cells, which you studied in F214, which we could transplant into the islet of Langerhans, they might be able to differentiate into beta cells that work in the way that the healthy beta cell would work. I detect glucose levels and release insulin, and there is some research, and there's been some success in animal models of doing this. There are problems with this. Obviously, stem cells, where do we get stem cells from? Embryonic stem cell research, there's obviously all sorts of ethical issues involved with that. But there is potential hope for the future that stem cells and transplanting stem cells into the pancreas could actually provide a cure, i.e. we wouldn't have to keep retreating the patient if we managed to transplant stem cells into the patient, which would produce insulin for the patient. Okay, so not presently available in humans, but a lot of research going on here, which could make this a cure for type 1 diabetes in the future. Anyway, that finishes our screencast on everything to do with the pancreas.